This is part 2 of our presentation on the cell. We had been talking about cytoplasmic organelles and had made it to the Golgi apparatus. A little bit about the Golgi apparatus. It modifies and packages proteins, and it produces different types of packages, including secretory vesicles, cell membrane components, and lysosomes. Now, the Golgi is sort of a multi-layer group of flattened sacs. And what happens is that proteins that are fabricated in the endoplasmic reticulum are moved to the Golgi apparatus, where they are modified and packaged and processed. I think of this kind of like factories. In the endoplasmic reticulum, what we have is a manufacturing plant. I worked at a GE plant, and they produced headlamp components. But we did not make headlamps. What we did was we shipped those components to a different factory, which put the components together, stuck a light bulb in, and made a finished headlamp. And that's pretty much what the Golgi does. It takes the components that it receives, processes them, and makes them functional. Other cytoplasmic organelles, we mentioned that the Golgi could make secretory vessels, cell membrane components, or lysosomes. So let's talk a little bit about lysosomes and actually another component, peroxisomes. Now, lysosomes are packets that contain digestive enzymes so that if the cell brings something into it, or if there's a component of the cell that is old and non-functional, they can be broken down. Those enzymes will break proteins down back to the constituent components of amino acids, and so on and so forth. Peroxisomes are a protective device that many cells have. They contain sacs of an oxidase enzyme, and an oxidase is the enzyme that breaks down peroxide. It destroys it so that it can't do harm. So these peroxisomes can detoxify some harmful substances. They break down free radicals, which are highly reactive chemicals that could damage the cell. They replicate on their own by pinching in two. The nucleus in this case does not guide the production of peroxisomes. When we pour peroxide on a wound and you see that area foam, what you're seeing is the reaction between the peroxide oxidase in the peroxisomes and the peroxide itself. The next organelle is mitochondria, and these are termed the powerhouses of the cells. The reason for this is that they can take a byproduct of glucose breakdown. Glucose breaks down into two molecules of something called pyruvic acid, and it can feed those into the mitochondria, and you end up getting 32 to 34 more ATP. Now, when you just break down glucose, you only get 2. Prokaryotes were not as efficient as eukaryotes. Prokaryotes do not have mitochondria. As a result, prokaryotes can only get those 2 ATP, which is the energy currency of the cell, and then it releases some kind of byproduct. Now, an example of a byproduct that is released by yeasts is alcohol under the right set of circumstances. Mitochondria carry out reactions where oxygen is used to break down food. So all of the air that you and I breathe in is used by the mitochondria to provide ATP. Inside cells, there's also the cytoskeleton. And the cytoskeleton is a network of proteins that extend through the cytoplasm, and they provide the cell with an internal framework. So in order to keep the cell in a certain shape or modify its shape, we have these cytoskeleton. The structures of the cytoskeleton also function as transport devices. They think of like a zip line. You basically have something that you want to go from point A to point B, you attach it to a thread of the cytoskeleton that goes from those 2 points, and then it gets carried to the desired location. Centrioles exist in cells that go through mitosis, again, eukaryotic cells. These are little rod-shaped structures. They are made up of very small microtubules, and they are necessary for the formation of the mitotic spindle, which is what draws the DNA or chromosomes to either side of the cell and divvies it up so that each new cell will have the same copy of information. A lot of cells have cellular projections. They're not found in all cells. And in microorganisms, we do see a lot of cilia and flagellum. Now, cilia are very short projections that are on the surface, and they have this waving motion that will allow the microorganism to swim. It's almost like you have hundreds of little oars. In eukaryotic cells, in organisms such as us, we do have cells that have cilia. The cilia in that case moves something else. It doesn't move the cell itself, but it wafts along and carries things such as mucus from our respiratory tract upward. Now, flagellum, these are longer whip-like structures, and in microorganisms you can have multiple flagella on microorganisms. In humans, what we see is that there's just one cell type that has a flagella, and that's the sperm, and it has a single one. But as we go into the bacteria, what we'll find is that there are a number of microorganisms that have flagella on one end, off the side, in the middle, off both ends, and we have names for each of those types of propulsion. Again, just to look at the plasma membrane, because we're going to talk about membrane transport. So when we look back at this membrane, it is a selectively permeable membrane. It separates the inside from the outside, but there are proteins that allow things to pass through. This protein doesn't have any kind of door or anything on it at all, so it just allows certain molecules to pass through at any time. This one can be opened and closed. You see there's like a little flap there, and it can allow certain substances to go in or out when it desires. Now, membrane transport, there are two basic methods. There's passive transport, which doesn't require any energy at all, and cells try to be efficient. So if it can use stuff as sort of a free ride, that's great. Free is much better. Active transport does require metabolic energy from the cell. In passive transport, what we have is material that's going from an area of high concentration to an area of low concentration. In active transport, generally what we're doing is we are pumping material from an area of low concentration to an area of high concentration. Substances tend to want to spread out, and so passive transport, again, harnesses the energy of them spreading out, allows the materials to either be absorbed or released, again with no energy involved. But when we have materials that are highly concentrated and we want to concentrate them more, that takes some work. And so we have a number of things that actually are protein pumps that will move materials and concentrate them. Now, protein synthesis. We went through this a little bit earlier on in chapter 4, but just to reiterate, the gene is a portion of DNA. that carries a blueprint for building a single protein. So when we think of a gene, it is the blueprint for a protein. And proteins have all sorts of functions in the cells. They are what make cells work. They are building materials for the cells. They act as enzymes, which are biological catalysts, so that reactions can happen much, much faster. We could take the materials in a cell and put them in a jar and shake them up, and we might have to wait 1,000 years for a reaction to occur. But with biological catalyst, it's a matter of milliseconds. Now, we talked about DNA. RNA is essential for protein production, and there are multiple types. There is transfer RNA, ribosomal RNA, and messenger RNA. Messenger RNA is taken from or copied from the DNA. And then the messenger RNA goes to the ribosomal RNA. It is the machinery that reads the message and builds the protein. And it does that with the help of transfer RNA. Now think of transfer RNA as being like a gopher on a job site. It goes and gets what the ribosomal RNA needs to build the protein based on what the message from the messenger RNA tells it. The process involved here, when we copy DNA to make the messenger RNA that can be read by the ribosomes, when we're copying DNA, that is called transcription, meaning to write. When we actually read that messenger RNA and are producing our protein, that process is called translation. This graphic pulls all of this together in one place and how each of the organelles comes into play. So what we have is DNA in the nucleus, which gets transcribed to messenger RNA. The messenger RNA exits the nucleus, where it is picked up by ribosomal RNA in the structure of ribosomes. The ribosomes read the message, and with the help of the transfer RNA, you get transfer RNA bringing the necessary amino acids and attaching them one at a time in a chain to form the primary structure of a polypeptide. Ultimately, that polypeptide, once it's completed, will be modified in the Golgi apparatus and be released either into the cell or released for export. This sums up the general information on the cells. Again, this will be helpful to you when we get into Unit 2, where we start talking about individual microorganisms. Thank you much.